Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Bacterial protein synthesis. Imagine you getting tetanus. Now, Salmonella tetani is an organism which can cause tetanus in you, or you are getting a bacterial infection which is an E. coli infection. Dysentery is a common symptom of food infection or food poisoning that's caused due to E. coli, and this E. coli is not the normal flora of your body. This has to be the causative agent of dysentery, which is a harmful bacteria. Now, how does that harmful bacteria cause a disease in you? This harmful bacteria will gain entry inside your body with the help of the food which you're eating, which is unhygienic or which is contaminated with either food or certain kinds of bacteria, which could be by water process or any such process. And once those E. coli species gain entry into your body, they will start the production of certain proteins, and those proteins would be responsible for causing disease in an individual, making a healthy individual unhealthy. Now, these proteins will only be synthesized if the whole process, that's DNA replication, transcription and translation, is all done in E. coli. So just to stop that, we are given certain medicines, and these medicines are antibiotics. And one of the 4 common antibiotics that inhibit the protein synthesis in bacteria and kill those bacteria and prevent us or cure us from that disease is tetracycline, streptomycin, chloramphenicol and neomycin. Now this all are antibiotics. When I say antibiotic, it means anti means against, and when I say biotic, it means against genetic organism, or you can say against organisms. Now antibiotics are nothing but medicines which counter attack your causative agent, or you can say the harmful E. coli species, and they somehow manage so the protein is not synthesized in your body. And if that protein and enzyme is not synthesized in your body, obviously there is no chance that you would land up with the disease. Now all of these enzymes work, or you can say all of these antibiotics work in different fashion, and they have different manner to counter attack the protein synthesis, which we'll be studying today. Now some of the inhibitors which function to stop the bacterial protein synthesis are tetracycline, streptomycin and chloramphenicol. Now how does tetracycline works to stop bacterial protein synthesis? Basically, your tetracycline inhibits the binding of amino acyl tRNA to ribosomes. Now amino acyl tRNA is nothing but activated transfer RNA. Now imagine a situation in which your activated tRNA cannot bind to the ribosomes. mRNA is already present with its genetic basis exposed. On that, your ribosomes comes and bind. Your tRNA, or you can say the transfer RNA, should basically carry activated amino acids from the cytoplasm and give it to the mRNA. And anticodon and codon bonding has to take place. Now if tRNA is blocked, do you all think so that anticodon and codon bonding will take place? Obviously not. And if that doesn't happen, no protein is synthesized, and hence the organism cannot cause any disease. The next one is streptomycin. Now streptomycin basically inhibits initiation of translocation and may cause misreading. Now, streptomycin works in the last phase of protein synthesis. What it does is, once your polypeptide chain is formed with all the peptide linkage and the protein is almost complete, it has to be translocated, or you can say transferred, to the site of action. Now, to do that, there is a machinery or mechanism that has to be followed. Streptomycin just makes that machinery or mechanism misleading, so that the protein cannot be transported to its actual site where the bacteria actually wants it to reach, so that it can cause disease to you. And this is the way streptomycin works as a medicine source. The third one, chloramphenicol. Now, chloramphenicol basically inhibits peptidyl transferase and the formation of peptide bonds. Whenever amino acids are joined together, the bond that is formed is a peptide bond, or you can say the CONH linkage. For that to happen, there's an enzyme which is called as peptidyl transferase needed. Now, your chloramphenicol basically blocks peptidyl transferase enzyme. So if there is no peptidyl transferase, we all know that the polypeptide chain that is joining the amino acids won't be formed. And if there is no polypeptide chain, there is no protein, and hence no action or no harmful effects of the bacteria. And lastly, the antibiotic neomycin. What is neomycin does is, it inhibits, or you can say it stops, the interaction between tRNA and mRNA. And we all know that mRNA is your messenger RNA, which will have genetic information for the protein synthesis, whereas your tRNA is the transfer RNA. So if the tRNA, which is the transfer RNA, could not carry the amino acids from the cytoplasm and bring it to the mRNA, which has the codon region, and there is no formation of codon-anticodon pairing, hence no proteins would be synthesized. And obviously, the mechanism by which bacteria is working to cause disease in an individual will come to a halt, or it could fail. So students, whenever you fall ill and it is a bacterial infection, all these antibiotics are given, and antibiotics basically have a vast effect, or you can say they have a drastic effect, because they not only treat the symptoms, but they also directly trigger, or they pinpoint to the leading cause. They directly kill to the bacteria. Antibiotics can be broad spectrum or narrow spectrum. Now, narrow spectrum antibiotics work on certain medicines or on certain symptoms, whereas broad spectrum antibiotics like rifampicin are given to tuberculosis patients which could have infections of many. So for a dysentery infection caused due to E. coli, which is a food source of E. coli which you have, which has been hampered with causative agent of E. coli that's poisonous to you, you will be given a narrow spectrum antibiotic. But if the disease is dangerous or this life threatening, that time broad spectrum antibiotics are given. I hope you all are clear with this concept. Thank you.